live on YouTube. I will ask just a few minutes for people start to enter in our channel in YouTube, because there is a little delay between what are we saying and viewing in the mconf and what is transmitting in YouTube.
Okay.
But yes, we are live and there is people entering in the channel already. So we'll start in a few minutes. Bom dia, pessoal. Eu vou dar mais um minuto ou dois para todo mundo conseguir entrar. E aí a gente pode iniciar. Okay, there are a lot of people already in the room, I guess. So, I guess we can start. Good morning, everybody. Today, our guest is Professor Judith Hollinger from University of Vienna in Austria. Thank you very much, Professor Hollinger, has accepted the invitation of Professor Eduardo to be here today. I will close my microphone and my camera to not interfere with your presentation, but I am listening here all your presentation. So anything you need, anything happen, you can call me.
Perfect. I hope you can hear me.
Yes, perfect.
Okay, then fine. Thanks a lot. It's a big pleasure. Thanks a lot, Professor Marcella Arbo and Leonardo Conrad, for this kind invitation. It's entitled Computational Methods in Natural Product Research. And I'm a professor at the Division of Pharmacognosy at the Department of Pharmaceutical Sciences at the Faculty of Life Science at the University of Vienna in Austria. And it's my big pleasure here to welcome you to this morning. Actually, it's afternoon here in Vienna with this topic. So what is it about? What you can see here on the slide and here in detail on the left side is actually ceiling paintings of one of the faculty paintings made by Gustav Klimt. And I could imagine that Gustav Klimt is also known in Brazil for his very famous art, famous paintings. Unfortunately, this building, which you can see here as a black-white photo, is not existing anymore as the original. It has been painted for the University of Vienna's Great Hall, and it's one of these faculty buildings. So for philosophy, medicine, and jurisprudence, he was responsible for painting it. However, when finalizing this picture and having it on the ceiling, Klimt came under massive attack for pornography and perverted excess in the painting's art, and therefore he had to remove it. It was one of the biggest scandals in the very beginning of the 20th century. And it was then stored in a castle, and this castle called Schloss Immendorf was destroyed in the very last months of the Second World War. So what we have is just a black and white photo from this very famous painting. And whenever you have the chance to be in Vienna, you might see a lot of umbrellas and cups and porcelain, or with these very famous Hygeia, which you can see here in this close-up. And usually you have depicted her in gold and red. However, this is just from memory, because the colors are not Of course, they are known, but we don't have a colored photograph of this painting. And Hygeia, as you might know, is a Greek goddess of health and cleanliness and sanitation. Therefore, very important for our profession. When we are looking at the time 100 years before, so not at the beginning of the 20th century, but at the very beginning of the 19th or even late 18th century, the term pharmacognosy was coined in Vienna, and it was coined by Johann Adam Schmidt in Vienna. And you see this building here on this photograph, and it's also the building which you can easily also recognize today. It looks identical. The streets, of course, changed. There are no horses anymore, and there are a lot of traffic. But nevertheless, the building itself didn't change a lot. And the name pharmacognosy was because Johann Adam Schmidt named it as the second discipline in pharmacy. So it's not only pharmacology, caring about the pharmacological and the biology of these natural compounds, but the knowledge about these drugs. And since these drugs at that time were all from natural sources, it's becoming the naming for our research field. So everything connected to research in natural products. Coming from the Greek term drug, pharmakon, and gnosis, the knowledge of these drugs. Yes, in 1795, just that you have a small historical background, Johann Adam Schmidt became professor at the Collegium Medico-Chirurgicum Josephinum, which is this building in Vienna, and it's still there, and you can visit it even today. It's storing a museum with wax models, which are very, very, very world famous. This is a painting of this professor, Johann Adam Schmidt. He was actually very much into ophthalmology and famous for cataract surgery and inflammation of the iris. But he also wrote a textbook. It's called Lehrbuch, which is a treatise, Der Materia Medica. And it was posthumously published. And it holds everything, all the knowledge at that time about natural products and their healing properties. And there, in this book, pharmacognosy was originally coined. By the way, Johann Adam Schmidt is also very well known as the personal physician of Ludwig van Beethoven, and therefore there are also some pieces of music dedicated to this very famous Johann Adam Schmidt. So that's about the background, the historical background and the name of pharmacognosy. What is it today? Today, it's the study of physical, chemical, biochemical, biological properties of drug substances, potential drug substances of natural origin, as well as the search for new drugs from natural sources. As you can see already from this description, it's highly interdisciplinary. And it's continuously evolving, and new approaches are coming in, new techniques, and therefore it's very, very timely, always changing. But what keeps the same is that it's dealing with natural products and their impact on health. So we have a lot of disciplines today, and what's more and more important is to connect all the knowledge we have about chemistry, but also biology of natural products. And with the aim to have finally effective and safe herbal remedies, which are quality controlled, and of course is also one of the main aims to discover new potential lead structures from natural sources. But where do we usually start with? There are different starting points depending on the disciplines we are working, and as you could see there, it's a broad field of natural products research and the disciplines within this research field. So information, a starting point could be from traditional medicine, we could also have some starting point from an extract screening or some chemotaxonomic relationship, simply serendipity, looking at what we find. And also the bioactivity readouts can be from very different areas. So it can be from an enzyme-based assay, or a target-based assay, can be a cell-based assay, or a phenotypic assay, or even from in vivo experiments, or even from long-standing application, which also falls into this traditional medicine and ethnopharmacological knowledge. But usually, we are starting with multi-component mixtures with no idea about their constituents, their fate in our body, their interactions to the targets, and finally, their impact on our body. So there are a lot of question marks. And I take a phrase from Voltaire, who said, doctors put drugs of which they know little into bodies, of which they know less, for diseases of which they know nothing. And if we translate this to pharmacognosy, it's that pharmacognostists examine extracts of which they know little, models of which they know, identify activities of which they know nothing. And fortunately, it's not that harsh anymore. And it's a very provocative statement of mine, but nevertheless, there is much room to further explore so that we have more insight into molecular mechanism, also about the fate of these natural compounds in our body and about their impact. So how can we harness complexity? And we have to deal with complexity whenever we are working with the extracts, for example, using combined approaches. And that brings me to research where we try to integrate big data analytics, in particular computational tools, combining it with experimental testing, but also with knowledge from traditional applications, so from ethnopharmacology. And I will show you some examples from my research lab and from my research group. It's about antimicrobial screening. And on the other hand, it's also about the HERG-related toxicity, so the cardiotoxicity. These are the two main examples which I want to give you today. The HERG channel blockers in medicinal plants. This was one of the initiatives some years ago. We wanted to know if it's worth to dig into it, if there is potentially a risk when consuming medicinal plants or products from medicinal plants. Is it hysteria or is it a serious problem? And this was funded by a EU project. It's called HERG Screen, and it's some years ago that we accomplished it. So as you can see, almost 10 years ago. I will bring this example because we also had some very, very nice collaborations with Brazilian partners and also with your university. The cardiac ventricular action potential. This is where we were looking at because these different stages, also called QT interval, corresponds to the duration of the ventricular systole. And there is the rapid delayed rectifier current through the HERG channel, which is important because it terminates the action potential. If this repolarization is through the HERG channel is inhibited, we suffer from pro-arrhythmic effects, and in very severe cases, we are suffering from so-called torsade de pointe, which might be fatal. So drug-induced QT interval prolongation is a significant hurdle in drug discovery, and this was already recognized and probed. Therefore, all the drug candidates are probed for this QT interval prolongation. In the ICH guideline, it's called the non-clinical evaluation of the potential for a delayed ventricular repolarization by human pharmaceuticals. So it's very clear that we have to care about this potential cardiotoxicity issue. However, this has until then never been probed with natural compounds or with herbal medicines. So our question at that time was, what about potentially herb blocking natural compounds? It's time to systematically explore natural compounds and to evaluate their potential cardiotoxic risk, and thereby to improve the safety of the consumers. And this was by establishing a network between European, South American, North American, and also South African educational and research entities. The aim was to identify potential risks by collecting or investigating commonly consumed botanicals in these different continents. Can you still hear me, or can you see?
Yes, I can hear, but the screen, it's not sharing anymore.
Okay, I share it again.
I don't know what happened.
Can you see it? One moment. I just try to fix it.
Yes.
Okay, fine. You can see it?
Yes, it's great.
Okay, then I continue.
Okay.
So what you see here is this network of the EU consortium. We had a lot of partners, and you can also see, by the way, can you see also the cursor? Can you see my pointer?
Yes, I can see your pointer.
Wonderful. Fine.
What?
It has gone again. I try it again. No, it doesn't want to take it.
No.
No, I try to share it. I
It's
also
sharing
see
again.
Yeah, I try it again.
It's working.
Yeah, here it is.
It closed it again.
Close it again. I can try to step out of this video call and come in again, probably.
Yes, it's possible.
I once tried again to share my window. Now
yes,
you should see
it's
it.
sharing. Yes.
Yeah. Yeah, let's go and try if it works. And
Okay.
when it crashed down again, I can also try to step out and step in again.
Okay. Okay.
So you can see here the partners of this EU project, and among these partners is also Porto Alegre and your university. It was Teresa de la Costa who did all the pharmacokinetics work in collaboration also with the group from Bloemfontein and also with the other partners. And there were a lot of secondments, and so a lot of early stage researchers and experienced researchers changed the continents and the countries, and there was a lot of experimental working together, training, teaching, and also a lot of workshops that have been organized. Here you can see some of the colleagues within this consortium. So I show you briefly how the workflow was established and how we proceeded in collecting the material, which led then also to the extracts and further on to the investigating these extracts. So we selected commonly used botanicals from 3 different continents, and finally we came up with 650 plant species, prepared extracts out of it, and these were then roughly 1,200 extracts. And on the other side, we generated based on, it's again closed.
Yes, close it again.
Okay.
I guess she'll try to get out and get in again. Não sei porque eu tô falando inglês, mas porque ela não tá aqui, mas eu acho que ela vai sair e vai entrar de novo. A transmissão tá ok aí, pessoal?
Ainda mais
Professor Hollinger?
Yeah.
Yeah, everything okay?
Yeah, from my side, everything is okay.
Okay, I will turn yourself again a presenter.
Yes.
Then let's see if work. Yes.
Okay.
I can
Give
see
it a further trial.
Yes.
Good. I closed my cameras, maybe then it's more stable.
Okay, no problem.
So let's continue. So we generated pharmacophore models. So based on information we already had from the three-dimensional structures of the protein side, so from this channel, from the binding sites of the channel, as well as from ligands at that time, we've generated a set of pharmacophore models. And this set, which is depicted here in the middle, we selected virtual hits by virtual screening of an in-house natural product database. And this was the first experimental evaluation if these predictions were correct, or if we can build up models or further experiments based on these pharmacophore models. So what we did is that those that were virtually predicted to interact on the channel, on the binding site of the channel, were in vitro tested in a Herc patch clamp assay in Vienna. On phytochem on the other side, it also guided us which extracts or which plant material could include constituents that show a certain propensity to interact on this ligand binding domain. We identified potentially cardiotoxic botanicals and also its constituents and validated it by in vitro assays, and further on the idea was also to do PK and PD studies. With this, we first did this, we compiled all the active constituents that were known as ligands. We created these common features. We selected the area which is restricted for the ligands. So we had some very nice ideas about the three-dimensional architecture of a ligand, which should be fulfilled whenever it comes into a Herc channel. With this, we predicted some natural compounds and experimentally validated it on HEC2 293 cells. And interestingly, 20 of the 50 selected virtual hits showed also a nice 50 value between 0.1 and 2.8 micromolar. So that was a clear tool that is useful to be used to further predict novel hERG blockers. And so we did by searching big, large molecular databases consisting of molecular and natural products entities, and screened them with these pharmacophore models that were validated in a nice manner, and came up with an alkaloid, which you can see here in the middle of the slide. It's an indole alkaloid. It's called vorkangine. And by experimental validation, we could also see that this vorkangine really shows an IC50 value of 0.3 micromolar. It's an iboga alkaloid from different species of the Apicynaceae family. For example, from Vorkanga africana, from Tabanante iboga, but also from Tabane montana africana and several other Apicynaceae species. So when we look at this Vorkanga africana, then we can see that it is used a lot in folk medicine, in particular in Africa, against different diseases. It is also used for religious purposes as a hallucinogenic drug and aphrodisiac agent, in particular the seeds and the bark of this tree, which are used. What we did is we bought some of these products because it was not forbidden. It showed at that time an increasing popularity as a legal high in Europe, with very exciting names, as you can see here on these pictures: herbs of the gods or seeds of the gods. And we investigated this bark material. The iboga alkaloid vorkangine is it's mainly used not only as hallucinogenic, but also as a precursor for the same synthesis of ibogaine. Here you see the differences. So the only difference is this carboxyl, this methyl carboxyl group at position 18, which differs between vorkangine and ibogaine. And ibogaine itself is used for the ability to reverse addiction to recreational drugs such as opiates as well as alcohol. It also shows some ability to reduce craving, to come up from if there is an addiction to any of these drugs. However, there are no controlled studies in humans, and there is no information or little information available on safety and efficacy data. In the US, it's a Schedule 1 controlled substance, and in many other countries, it's almost unregulated or unlicensed. So there is a question, there was our question, what about the in vivo relevance of vorkangines in vitro data? Can we really say, because of our experiments, that it might be cardiotoxic? And therefore, we first had a look into this natural material, actually the wood bark of Vorkanga africana, and could identify vorkangine in very high yields. So it's almost there in 10% of the crude extract. And we did a preclinical PK study together with Teresa de la Costa. And this was done in rats, divided into 4 groups with 5 milligram per kilogram IV of vorkangine, which better than 5 milligram per kilogram rats and 50 milligram per kilogram rat, as well as in comparison with the crude extract in an equivalent. What we saw from this PK study is that the oral bioavailability is quite low. It's about 12%. And therefore, the question was, is there a serious risk when taking vorkangine or even ibogaine? It shows a relatively low bioavailability. However, it also showed a high plasma protein binding, a high lipophilic character because of the chemical structure. And therefore, it's also possible that it is deposited in fat tissue. So in some we could say it might not be a big issue if it's a healthy person, but if it's tremendously used, it could be really cardiotoxic, have a cardiotoxic effect, in particular if it's accumulated in fat tissues. When looking at all the botanicals in this EU project that were collected, you see that a lot of these botanicals are coming from South Africa, from our partner P9, but also a lot of extracts or plant species were gathered and extracted from Brazil. Actually, we worked together with Eloy Schenkel's group in Florianopolis and also with Teresa de la Costa in Porto Alegre. And the other partners contributed their species from different organs from the Mediterranean area or from the Alpine area. In some, we had 650 plant species, already mentioned, 1,200 extracts. This was screened in a 2 micro electrode voltage clamp assay on Xenopus oocytes. And we considered an inhibition of higher than 30% at 100 microgram per ml as an activity threshold. So if we use this threshold, out of these 1,200 extracts, 25, we can say only 25, showed a considerable inhibition or blockage of the hERG channel. And all these species were from 19 different families. So in some we could say maybe the commonly used botanicals are not so risky when it comes to cardiotoxicity. If you just look at the Austrian extracts, those that we collected in our lab, which is based on 2 issues. The one was that we screen very often used consumer botanicals. And the other issue was that we select those that were predicted to have a virtual hit inside. And if I take this red line as the threshold, you can see that out of the 61 extracts, 5 of them exceeded the threshold. And all of these 5, or 4 of these 5, actually to be concrete and correct, 4 out of these were predicted by virtual screening. The one is more familiar to you than it is in Austria, but nevertheless, it holds, in some monographs, in some pharmacopoeia, it holds a monograph, and this is Carapichea ipecacuana roots. The reason why we selected this was also because of our pharmacophore-based virtual screening, using a natural product database consisting of more than 120,000 structures, and the hit rate was roughly 0.8%. And out of this virtual hit list, we selected this Carapichea ipecacuana because these ipecac alkaloids deemed or showed a very nice ligand-target interaction. If I say very nice, it's not correct, but it showed that there might be a potential risk. So we investigated it and we isolated 5 ipecac alkaloids out of this root material, namely emetine, cephalin, psychotrin, O-methylpsychotrin, and N-methyl-emetine. And you see here also the IC50 values. Particularly it's cephalin, which showed the most dangerous inhibition of the hERG channel, but also emetine. Is this of relevance? It might be. What's the impact on human health? The ipecac syrup is actually a renowned emetic medication. It contains roughly 1 to 1.5 milligram per milliliter of total ether-soluble alkaloids. And this is mainly emetine and cephalin. And to be honest, there is a very, very high misuse of this ipecac syrup. It's not available in drugstores anymore, at least in Europe not anymore, but you can easily buy it in the internet. And this is used from patients with eating disorders. There is often a chronic abuse of ipecac syrup. And what we know is that these alkaloids are rapidly increased in plasma level, or there are high plasma levels of these alkaloids. They show a high tissue accumulation and they induce cardiac toxicity. But up to this study, it wasn't known from where the cardiac toxicity is coming from. So with this study, we had a clear idea that it is based on a hERG channel inhibition. Looking at one of the other hERG inhibitors, we saw that Nelumbo nucifera, the lotus leaves, also were predicted to show an interaction on the hERG channel. So we also looked at this plant material, at the lotus leaves, and isolated 5 alkaloids, 4 from the group of the aporphines, and 1 isoquinoline alkaloid, which you can see here on the screen. And when we identified their IC50 values, you can show that some of them really have micromolar, they are in the micromolar range. Again, the question, is this of relevance? Maybe you know that there's a so-called lotus leaf diet. It's a Hollywood starlet diet. Use a lot of botanicals, food supplements, lotus leaf in order to decrease weight. Nuciferine, one of these alkaloids, is known to block the fat absorption. And here you can see the advertisement of these lotus leaf, the pure concentrated lotus leaves. This nuciferine inhibits the pancreatic lipase and fat accumulation. At least it's reported from in vitro experiments, and it inhibits adipocyte differentiation. What we did is that we collected 5 different lotus dietary products from the US market and quantified those alkaloids which we have determined as in vitro hERG blockers. What we can see here, these colored rows are the 5 different products. What you can see here is that the difference in alkaloids as constituents in these products is very, very different. So finally, you never ever know when just grasping a product out of the shelf, you never know if you consume a high alkaloid content product, or maybe like this Nelmuts 4, a product where there is nothing inside. But in this case, probably it's better to have nothing inside, because then you don't risk to have a cardiotoxic effect. Here are the parameters for the quantification which we performed, and also the different products here are color-coded. And what you can see is that in particular the nuciferine and all non-nuciferine show very different concentrations in these products. And it's particularly those constituents which also show these in vitro hERG blocking properties. What about the pharmacokinetic studies? Here we could refer to literature data, and it shows that they also show a rapid distribution, an extensive tissue uptake, a poor absorption into the systemic circulation with an absolute bioavailability of 4%. It's distributed into brain, liver, and adipose tissue. So, the one caveat is that lotus products on the market are not standardized dietary supplements. You never know what you get. And the other thing is that we actually, from these results, we don't know exactly if there is any life-threatening experience if you use either a lotus leaf diet, if you use the anti-addictive hallucinogenic drugs containing guacanjine, or if you use the vomiting-inducing ipecac syrup. You never know it. But what we can say is that from all these 1,200 extracts which we probed in this project, roughly 2%, and only 2%, showed a reduction of peak trail current of 30% or higher per ml. Thus, there might be a very low risk for hERG channel-related toxicities by botanicals. However, these are only in vitro results. And of course, for a direct translation it needs further in vivo studies. But we can, at least from these results, we can say that for healthy people without additional medication, it seems not to be very life-threatening. However, you have to consider that if it's anyway used in continuously, chronically, excessively, and also together with other drugs, it might multiply the cardiotoxic burden. And this should be aware, so consumers have to be aware of it. So I here introduce those that participated in this project, which is Ulrike Grinke. She's still working as a senior scientist in my group. Christina Meyer, she did her PhD in this project. Shadel Müller-Kratz from Florianopolis. He's still back in Florianopolis, and this whole work was financed by the EU. And this was my first example. And if you like, I can go to the second example, which is the search for natural products against acute respiratory infections, which is, of course, a very hot topic, in particular due to the pandemics. And this is financed by the Austrian Science Fund. And again, here you can see those people who mainly contributed to this study. This is again Ulrike, Benjamin Kirchweger, and Andreas Vasilevich. Benjamin is a postdoc and Andreas Vasilevich is a predoc, so a PhD student. What we can see in previous studies is that there is a lethal interplay between viruses and bacteria. And here a photograph of the Spanish flu time. And if you look at the data, there are roughly 40 million death toll 100 years ago, more than 100 years ago, due to the Spanish flu. But intriguingly, the death toll was mainly due to a superinfection. 95% died because of a bacterial pneumonia, so a co-infection. And this is similar also in the other pandemics, not only with Streptococcus pneumonia, but also with Staphylococcus aureus and also other bacteria. So in a previous screening, we looked at traditional medicine, traditional products that have been used whenever it comes to acute respiratory infections. That's why RE is standing for. And we identified a plant material which is in Chinese called Sangbai Pi. It's the Morus alba root bark. You see the Morus root, not the mulberry tree, and this is the root bark here on the right side. We isolated the constituents and also did a structure-activity relationship, looking in particular at congeners of prenulated flavonoids. And some of them we depicted for further investigation. What we can see is we looked at dual inhibitors, so inhibitors of the virus, of the anti-influenza virus on the one side, but also inhibitors of pneumococcal growth. What you can see here on these electron microscopic pictures is the biofilm without an inhibitor. On the photograph B, you see the influence of oseltamivir. And on the right side, you see that compound 27, which has this very nice anti-pneumococcal and anti-influenza virus activity. And you can easily see that even with a strong antiviral neuraminidase inhibitor like oseltamivir, nothing disrupts this pneumococcal biofilm. However, with a dual inhibitor, you can disrupt the biofilm. This was one of the starting points. And then in the pandemics, we were looking at implementing computational approaches to further look for antiviral and antibacterial natural products, using cheminformatic tools, but also combining it with a phenotypic extract screening and big data analytics. In particular, we used biochemistry, molecular networks, but also molecular modeling, docking, and virtual screening. And this time we not only looked for influenza or anti-influenza activity and anti-pneumococcal activity, but also for compounds acting against SARS-CoV-2. Indeed, for the virtual screening, we focused on two proteases. These are the main protease as well as the papain-like protease. Both are essential for the virus for replication. And thus, we use the molecular docking approach. This worked like the following. We selected PDB entries of either the main protein, a protease with a ligand co-crystallized, or the papain-like protein. And here you see the PDB entries for these protein structures. Having this structure information, we use the included ligands for a self-docking to show if it works. In parallel, we prepared all the molecules from two databases. On the one side, the in-house database. This consists of natural compounds we already isolated all the time, and 1,300 compounds. And on the other side, with a MolPort natural compound database consisting of molecules, so not physically available, but the majority of a big part are available to buy. And this is the MolPort natural compound database consisting of roughly 140,000 compounds. These were subjected for virtual screening with this molecular docking approach, and we looked at 15 different poses. We did a visual inspection of the highly scored docking poses. We looked thereby for binding interactions, for binding site placement, for pose reproduction, for docking scores, and selected virtual hits out of that. Finally, we selected 60 virtual hits for these 3 different PDB entries. And from these 60, 35 were available either by isolation, or we had it in stock, or we could easily buy it. 35 of these virtual hits we selected for experimental validation. This was done with enzymatic assays for MPro and PLPro, so for the main protease and the papain-like protease. And we came up with a hit rate, a real hit rate, not only virtual hit rate, of 23%. That means we select an enzyme inhibition of more than 40% at 20 micromolar, we labeled as active. These 8 experimentally confirmed hits were very precious for us, but we were questioning ourselves, are they really able also to protect from SARS-CoV-2? We know that they can inhibit these enzymes, but we don't know if they can protect from viral infection. So these are the 8 experimentally confirmed virtual hits, which were then in collaboration with the colleagues in Frankfurt in Germany submitted to a phenotypic assay where CALU3 cells were infected with SARS-CoV-2. And what you can see here is that in particular 3 of them could inhibit the SARS-CoV-2 from, could protect the cells from SARS-CoV-2 infection. And if you look at the structures and also at the names, it was very intriguing for us because these are compounds from mulberry tree. And the mulberry tree was already mentioned before because it's traditionally used against symptoms of acute respiratory infections. Looking at, for example, the sangenone, by the way, these compounds consist of a flavonoid unit, as you can see here on the right side. On the other side, you have a chalcone unit here. And via Diels-Alder adduct, forming the cyclohexane ring, due to this isoprenyl or doubled isoprenyl, actually geranyl moiety, a Diels-Alder adduct is formed. So these sanginones are also called as Morus Diels-Alder adducts. We had a very clear idea how it would fit into the main protease, but of course this is just hypothesis. And when looking at the ligand co-crystal structure And if you look at the docking pose of this sangenone G, you see that it nicely fits into this binding cavity. And if you compare it, it's quite similar than a butterfly with the 4 wings. And this is quite identical from the experimentally confirmed chemical X77, which is co-crystallized, as well as the docked sangenone G from mulberry root. This also refers to the other sangenones which we isolated from this material. They all looked very similar when it comes to the docking pose, but still this is a hypothesis, and we wanted to know to have clarity if this is the truth. We were not able to co-crystallize it, but we did STD NMR studies, so saturation transfer difference NMR studies, and with this so this is not depicted here, you can easily see that exactly those interaction points are confirmed by STD NMR. So in sum, we could show that starting from a 2D library, transforming into a 3D library, and the virtual hits, virtual screening, we could identify some virtual hits. We then did the enzyme-based assay for experimental assessment, and those that were active there we subjected to phenotypic validation, and also coming up then with compounds showing an antiviral effect. This was just recently published in Journal of Natural Products, and we are very happy about that, was selected with the ACS editor's choice. So we could identify new lead structures for drug discovery, but we could also rationalize the traditional applied herbal remedy sung by P. In Chinese, it says, in Chinese laws, it says, sung by P drains the lungs, especially heat in the lungs, thereby alleviating cough and wheezing. So symptoms that really also perfectly match not only with the symptoms from SARS-CoV-2, but also with other symptoms of respiratory infections. But what does it mean? What implications does it have? Which, if we want to go and bring it further in a translational work to the patient, which source is meaningful? Shall we go for isolating the compounds? Shall we go for synthesis, semi-synthesis, or with enriched extracts? What we know from enriched extracts is that there are some main contributors to the effect, but the mixture of sangenones in the plant material has a very nice additive effect. In sum, we know that the isolated sangenones show the in vitro activity against influenza viruses and different strains, or even from influenza viruses against Streptococcus pneumonia, as well as SARS-CoV-2. But which morus alba sample is best suited for future preclinical studies? Is it the leaves, the twigs, the root bark, or even the fruits? All 4 of them hold a monograph in the Chinese Pharmacopoeia. We wanted to know which organ provides us with the highest content of sangenones. Which is the best extraction method? Is it worth to use such a multi-component mixture? Or shall we go for isolated or synthesized compounds? Therefore, it was another PhD student, Julia Langeder, as well as Ulrike Grinke, who cared about different extraction methods from the different organs, using pressurized liquid extraction, traditional extraction, like a decoction and infusion, but also supercritical fluid extraction with CO2. And we could very rapidly see, when analyzing the sangenones from all these extracts which were gained from all the organs from different extraction protocols, that only the root barks contains these mulberry Diels alder adducts, NDAAs. So these sangenones, we can see here different sangenones. even it's not depicted here exactly, but you can see that they also hold different stereochemical centers, and depending on the stereochemical centers, you have different peaks here, like 2, 5, and 7. In sum, Julia did a quantification of all 7 sangenols in 65 extracts. Whereas Michaela Schmidtke from the University Clinic in Vienna, she cared about the antiviral testing. And what we did is that we looked at all these extracts from the root bark and identified quantified the content of these Mulberry-Diels-Alder adducts. And you can see, if we use a pressurized liquid extraction, we could gain up to 30% of the sangenols, with traditional extraction up to 6%, and with supercritical fluid extraction up to 20%. What we did then as a next step was that we looked at, on the one side, the cytotoxicity in NDCK cells at the minimal inhibitory concentration determined against Streptococcus pneumoniae, so the anti-pneumococcal activity, but also the anti-influenza virus activity. And what you can see here is just like a traffic light. Whenever the traffic lights were green, these were the perfect extracts for us. And this is also depicted here on this graph. So what you can see on the y-axis is the inhibition of influenza virus-induced cytopathic effect. So the higher, the better. And on the other side, on the X scale, you see the content of the sangenols, so the Mulberry-Diels-Alder adducts. And what you can imagine here is a kind of correlation. Further on, you see red, yellow, and green balls represented the color code of anti-pneumococcal activity, with green being the most active ones against pneumococci. So we selected out of these extracts, we selected this MA60 because we wanted to have an extract, a well-defined extract for further in vitro and in vivo studies. And it was then the next step that we wanted to combine it with a biochemometric approach. That means we used the ELINA heterocovariance. That means that we subjected all 26 pressurized liquid extracts to HNMR and correlated it with the activity with the anti-influenza activity. And by combining the spectral data with bioactivity data, we could create a pseudospectrum, which is nothing else than big data analytics using this combination, this connection between chemical features from NMR, in this case HNMR, with bioactivity data. And looking at this heterocovariance analysis, you see, like in a heat map, that those signals depicted in red correlate with a high activity, those in blue correlating with a detrimental activity or low or non-activity. And by this correlation, you can easily depict those signals that belongs to these molecules, or let's say chemical characteristics that belongs to those molecules that correlate with activities. And this way, we could identify sangenol C as the main contributor to the anti-influenza virus activity. This, our favorite or hit extract candidate was then also tested not only against oseltamivir resistant strains in different concentration, but also oseltamivir susceptible strains. And in all of them, we could see a dose-dependent inhibition. As well as we could see the inhibition was due to the inactivation of viral particles, preventing the binding to the host cells, which you can see primarily in this broad field here, pre-treatment of virus having an effect. However, pre-treatment of cells with mulberry root bark doesn't have an effect. High effect do we also have during virus adsorption, when the material is given during viral adsorption. So during and after virus adsorption, this is what we could see from the time-dependent plaque production experiments. So this brings me to the end of my presentation. We could see that this, what you can see here, which is nothing else than a modern translation of the Chinese application of mulberry root bark, saying that it's used against cough, against fever, against lung diseases. And we particularly looked at this upper part of the symptoms. And maybe in the next step, it would be interesting to look also at the other part of the body. But in this case, where we could see an effect on symptoms related to acute respiratory infection, there is a rationale that mulberry root bark extracts are useful. We could identify the bioactive constituents. We could also unravel the molecular mechanism. We know from this optimized extract that it has in vitro activity against influenza viruses, against staphylococci. We also know about pneumococci, against SARS-CoV-2, and it shows no cytotoxicity in lung epithelial cells. What we are currently doing is in vivo trials. We do have first in vivo trials where we have the doses, where we adapted to dosage. From the first in vivo trials, we can say that the bioavailability of these sungenolins is relatively low. So we are looking currently at the best application form. In particular, inhalation application form is currently in the focus of our research. And with this, I thank all those group members here depicted. We call ourselves the Phytochemistry and Biodiscovery Group, which is a fabulous group, and it's particularly those highlighted in red who contributed to the results of the studies which I showed you today. And of course, we are very happy that Mother Nature biosynthesizes these compounds. I also thank the collaboration partners caring about the antiviral testing. And I thank you, Eduardo, and I also thank you, Marcello, for the kind invitation and the introduction, and the funding bodies for financing our studies. And thank you all for your attention.
Thank you, Professor Hollinger, for your excellent speak. Then we have some minutes to open for questions and for discussion. Pessoal, no chat do YouTube, vocês podem fazer perguntas, comentários e observações. There is a little delay, as I asked before, between the YouTube and what are we chatting and listening and seeing now, so we can wait for some minutes.
Maybe I could be the first.
Yes, you could try.
Okay. I would like to thank you, Professor Judith, for your exciting talk. It's very nice to hear from your results, from your investigations, and I was also very excited for everything have you showed for us. And I think I would have two questions
Please.
for you. The first one, in your opinion, which could be the best to find some new bioactive molecules? Either if you could search for different ligands from a library of molecules and then search these compounds, perform a virtual screening, and then search for medicinal plants that contain these compounds and quantify them. Or if you could work with specific medicinal plants, then identify these compounds and then test these compounds in the virtual screening. Which would be the best option for to find some bioactive molecules?
Yeah, that's a very intelligent question, because actually, you could go for both directions. We have done all the directions, and of course, some are more successful than the others. It is very strongly depending on the information you have in advance. So if a druggable target protein is well known and you have a lot of information beforehand, such as crystal structure, a lot of ligands, so you know quite exactly how this ligand-target interaction is working. The more you know, the more accurate is the prediction. And then to say, okay, let's start with a natural product database, or it can also be a database with containing synthetic compounds, doesn't matter. And then to scale down those that have this three-dimensional architecture that fits into this binding site is a very legitimate procedure. Some bottleneck might be the availability of these compounds. So if you work together with collaboration partners from synthesis and they have large databases with synthetic compounds, it's fine because they can go to the shelf and say, okay, let's go and hand out those compounds that might be of interest for you. However, if you have to start from the beginning and to isolate these compounds that have been predicted, then it's sometimes time-consuming. It's sometimes also disappointing because if you have a hit rate of, let's say, a real hit rate, not virtual hit rate, but a real hit rate of 10% from your virtually predicted compounds, and you have to isolate 9 or 10 compounds before you approach an active one, it might be disappointing. So if you have already a physically available database, I think it's a good procedure to start with a virtual screening. On the other side, it's also highly interesting to first look at bioactive extracts, and then to say what metabolites, what secondary metabolites are inside this extract, which you could do by LC-MS dereplication. And then to say, okay, I might have these metabolites. Let's check which of these metabolites might interact to a target which is related to my assay. And then to say, this or that could fit into this model, and then to go for a targeted isolation. So this is also a possibility.
Yeah, I think we many times we start with this last option. We start working with bioactive extract, plant extract, and then try to search the compounds and see which compound is related with the activity. And
Yes.
then to perform a better correlation, we try to do a virtual screening to fit this activity to the individual molecule, which is also very possible to do. Yeah.
It's also depending on the assay you have. So if you have, let's say, any kind of antibacterial or antiviral or anti-inflammatory assay, and you can identify the compound which is responsible for this effect, and you want to know the molecular mechanism, this could also be an option to say, okay, I have the compound. I know that this is, let's say, anti-inflammatory, but I have no clue about the exact mode of action. So then you could also say, okay, taking this molecule and checking, for example, by similarity search, which means that you compare the 3D architecture of the molecule with a whole set of data, for example, from Kembe, to look at those that are already known as ligands of specific drugs or proteins. So you can say the more similar it is to a ligand, which is already known, the more the more likely it is that it really also interacts on that target. This is also a possibility.
Okay, yeah, that's very nice. I have another question. I have just made some notes here about ERG blocking natural compounds. I would ask you if there is a specific pattern or chemical core for these blocking compounds. All I have seen were alkaloids. All of them. Is there a specific class of alkaloids or a correlation that makes more sense to block this specific receptor?
Yes, that was a very good question. This was also one of our ideas when looking at these compounds, since if you build a pharmacophore model, we could easily see that there has to be an ionizable interaction, so a nitrogen. And we could clearly see that with all the virtual hits we identified, we are in the area of alkaloids. Otherwise, we don't have this ionizable interaction point. But it's not only this. It's also necessary to have a certain core structure, and it's also necessary to have a kind of linker which steps into, like in a doorway, to open up and to avoid to close the door. If we have these chemicals features, then we could see that these are some kind of requirements for these alkaloids. If you looked at the different alkaloid types we had, so they are quite diverse. Nevertheless, we tried to follow up with a kind of structure-activity relationship. What is necessary in terms of alkaloids to fulfill their criteria to act as a blocker of the hERG channel? And we did a summary of this and wrote a report on that, which is published in Natural Product Reports. And if you're interested, I can send it to you, where we try to follow up a structure-activity relationship. But it's not trivial. It's not that easy. Sometimes it's just a methyl group which inhibits the ionization on the nitrogen, and the activity is gone.
Okay. Oh, please send me this article.
Yes, I will do immediately after that.
To read about it.
Yeah.
Okay. That was all I had to ask.
Okay.
Okay, thank you so much. In the chat of the YouTube, there are a lot of people thanking you for the presentation. A lot of compliments.
Thank you. It was my pleasure.
Thank you. So I guess we don't have enough time for more discussion right now. Then I would like to thank you, Professor Hollinger, again for your speech and your disponibility to be here with us, at least online. Thank you, everyone. And I will speak in Portuguese now. Obrigado, pessoal. Esse é o último seminário, então, deste semestre. Encerramos hoje e retornamos à agenda em início de setembro. Provavelmente vocês vão receber por e-mail, no Instagram e nos outros locais as datas e os próximos seminários a partir de setembro. Então, obrigado. Eu vou encerrar a transmissão.
Thank you, Professor.
Thanks and goodbye to all of you.
I ended the transmission in YouTube.